Um, it is my pleasure to introduce Dr. Peter Park. He was originally trained in applied mathematics from Harvard and Caltech, and now leads a diverse group of computational biologists at Harvard University to study the human genome with applications to cancer. Dr. Park's recent paper describes musical mutational signature calculator. So I'd be interested to hear not only about the algorithm and how his team derived equations, but how they derived its name as well. Please join me in welcoming Dr. Peter Park.
Wonderful to be here. Can people hear me okay? All right. So the last time I was here was many, many years ago when June invited me. And so I'm glad to be back again. It's particularly gratifying to be speaking in the department that Gil is part of, because I met Gil many times when he visited Harvard. And every time I meet with him, I have to thank him because he was instrumental in my department of, I didn't write here, but Department of Biomedical Informatics at Harvard Medical School. So when Harvard Medical School was trying to decide whether to start my department, they asked Gil for advice, and he said, you should start the department. And so that was almost a decade ago, and I'm very thankful again, Gil. Okay. All right, so I'm going to tell you about some of the things that my lab is doing. And I'll talk about mutational signatures, but before I get there, I'll just give you some overview of my lab does, and then the things that I won't really focus on. And then I'll tell you a little bit of the introduction to mutational signature. I understand that most of you probably are not, maybe have heard of mutational signatures, but don't know the details. And then I'll tell you one story about how I have spent a lot of my time and energy in trying to take it to the clinic and how difficult it is. Okay, so not just the successes, but also the frustrating part of all this. And then at the end, hopefully I'll have time to tell you about this fun paper that I read about research productivity. Okay, so my lab is broadly interested in cancer and other genomes where we are trying to identify all sorts of mutations. So my lab was part of the TCGA project and spent a lot of time developing algorithms for cancer genomics. Well, it turns out that that was really a great setup for this next phase in my lab where we have been trying to identify mutations in non-cancer cells, right, where it's more difficult, okay? And so most of you know that you can sequence whole genomes and then identify not just point mutations, but all sorts of mutations, right? So point mutation is just one aspect of it that most people focus on. Copy number, there are a lot of experts here, but other types of structure variations that we just could not interrogate until we had whole genome data, we have tried to develop methods for. Okay, so that was from a review paper that we wrote recently. Now, one way in which we have expanded our genomics whole genome approach was to do, as I said, sequencing of non-cancer cells where the mutations are much rarer, right? So it's more difficult. There are a couple of ways to approach this problem. One is just to throw money at the problem, just sequence really deep. So we've done whole genomes of these brain tissues at 200, 300x coverage, right? So each assay was, $10,000, okay? Now, you can only go so far in that direction, and money cannot solve this problem because some of our mutations might be in 1% of cells, maybe even 0.001% of cells. Most mutations are actually private to a single cell, and you can never sequence your way into that, right? And so the other approach that we've taken is to do single cell amplification and to basically try to wade through the noise that you get from amplification artifacts to find mutations that are specific to single cells. And so we've done single cell DNA sequencing. So I didn't actually do single cell RNA sequencing, partly because my former postdoc was working on it and I didn't want to overlap with him. And so I just focused on single cell DNA. And so we can, as I said, do bulk sequencing at high coverage And that actually requires, because we're talking about 1%, most cancer algorithms, we're talking, you don't even test for variance under 5%, but we're really trying to push that limit. Or we do clonal expansion of single cells into bulk and then sequence, or the approach that we've taken, because sometimes it's hard to expand clonally a cell population, isolate neurons or other cell types, and then do single cell sequencing. So I'm not going to talk about this other than to say that this idea of identifying mutations that are present private to a single cell can have lots of different applications. And so, for instance, CRISPR experiment, right? When we talk about applying CRISPR for clinical applications, you have to know whether there are off-target mutations. Well, it turns out that unless you sequence single cells, you can't figure out whether CRISPR experiment is generating off-target mutations, right? How would you know unless they clonally expand, okay? Sequencing very high coverage exomes or genomes, you can't tell, right? So you have to sequence single cells. In fact, we actually find lots of mutations from CRISPR editing or even base editors. The latest base editors, they still generate point mutations that you cannot identify. We've sequenced mice that have been in space for a long time. We wanted to see if radiation in space flight would, or spending months in space would generate mutations and so on. So lots of applications of that technology. Another project that I just want to mention briefly is this SMART project. Some of you, just from talking with some of you during the day, I thought it'd be interesting to find out, for you to find out that there are projects like this where we're now trying to sequence normal individuals, multiple tissues, and using the latest technologies. So we're talking about, very high coverage, not just Illumina, but PacBio, ultra-long ONT sequencing, RNA using PacBio long reads, all sorts of things. So this is just in its second year, and it's going to generate lots of data that you should be able to access shortly. Okay, now coming back to the main topic, which is mutational signatures. Let me just introduce you the idea, and then hopefully try to tell you some problems and some solutions. So we know that different cancer types have different types of substitutions. And so if you look at lung cancers, you see lots of C to A substitutions and C to T substitutions. If you look at melanomas, it's mostly C to T, okay? So here I'm just showing 6 different types of mutations, because when you see, for instance, C to T, that's like G to A, if you look at the other strand, but you can't tell, right, which one is which. So we just narrow this down to the 6 possible cases. So the fact that different cancer types show different patterns of substitution is important, but it turns out that the context, the neighboring base pairs are also important. And so here, melanoma and glioma have the same C to T dominant mutation pattern, but you can actually look at the neighboring base pairs. Okay, so here I'm looking at the 6 substitution types plus 4 bases on the left and 4 bases on the right. So I have a vector of 96 that I can use to describe the context of mutation. It turns out that these two have different patterns, right? The C to T is there, but the contexts are different. So in the brain cancer, you have spontaneous deamination of 5-methylcytosine, which manifests in this pattern below, right? So you have this particular pattern because those are the contexts in which that occurs. For melanomas, we know that UV generates lots of CC to TT substitutions, and so that manifests in that pattern that's shown, right? So we can tell the difference between the tissue, okay, because they are exposed to different mutagenic processes. And so what we're trying to do is take a look at any sample, look at the trinucleotide context of the mutations, and then try to infer the process, okay? And so it's a little complicated because it's not like there's only one process that goes on in a tissue, right? There are multiple processes, and they act at different times to different degree, okay? So the mathematical question is, can we sort of undo this process and identify at the final stage, okay, given the mutations that you see, try to figure out what processes they were. Okay, so these might be three processes going on, and what you observe is some combination of those that have accumulated over time, okay? So from the observation, could we tell that there are three processes and what they look like? Of course not, you can't do it. But if you have enough data sets, if enough samples, then we can try to do this, okay? It's like listening to a band, okay? There are different instruments, okay? And if you listen to the music long enough, can you tell which instruments are present, okay? Well, if you listen to it for 10 seconds, maybe it's difficult. If you listen for 2 hours, maybe you can, okay? How many instruments are there? What are their different compositions at different times? And that's exactly the game that we play. The way to do this is something called non-negative matrix factorization. Some of you have taken classes where you learn about matrix decompositions and SVDs and PCAs. NFF is another one of those popular ones. It was published 25 years ago, has been cited a lot. And the ideas were actually relatively simple. It's some iterative, relatively simple iterative matrix factorization. And here I'm showing you how this differs from PCA, right? Most of you probably know PCA. And so this is the original image. The question is, how do we decompose that data into some components? And so decomposition by PCA is here, okay? And these are the weights of these different components, okay? And the difference between the PCA and an NMF is that, as the name suggests, non-negative, right? Every component here, all the weights have to be positive, okay? In mutational signature analysis, it's this additive effect of different processes. You can't subtract a process, right? And so you can see that it's dividing this image into different components, right? And so the reconstructed images are somewhat similar, okay? It's probably a little bit better with PCA because of this constraint, but we can, in fact, perform this decomposition. And that's exactly what we do, okay? And so the game that we've been playing is take all the samples that we can find, and here in this 2013 paper from Sanger, they've collected 500 whole genomes at 6,500 exomes, decomposed the matrix into components that make up this set of samples. And in that case, what they found was these different components, right? So mathematically, it's an interesting question how many components there are, but this is what they found. Since then, there's another version, 6,400 whole genomes and 19,000 exomes. You get another catalog, and you can actually do this with double base substitutions, indels, and then more recently, just in the past couple of years, copy number and structure variations. Okay, so right now, there is a database called Cosmic, okay, that you can go to, and there are dozens of signatures, and now we're trying to figure out what each signature means, okay? I mean, if there's actually a distinct process that we identified, it must correspond to a mechanism, okay, that generates it, right? Like the UV signature, right? Okay, so here are the dots represent the types of mechanisms that people have annotated, okay? And you can see that a lot of these have dots associated with them. That means we actually could figure out what it was. Others without the dots means we don't know. Okay? It appears to be a separate mechanism, but we don't know what it is. Now, how do we know these things? Well, we can do experiments, right? You delete some, polymerase component and sequence the whole genome, and then you see what it is. Okay, and so there's many efforts to figure out what these components are. Okay. All right, so there are lots of questions associated with this. I'll touch some of them. How many signatures are there, right? because we want to separate them into the right components, but not too many, right? Because we don't want to start subdividing. And I'll show you a little bit later how, as you accumulate more data, the number of signatures increases. And so the catalog keeps changing. More data, more signatures. Is that true? What is the mechanism behind its signature? The Cancer Research UK has just announced another set of questions under their grand challenges in cancer. And one of them is trying to figure out what every signature corresponds to. Okay? What is the best way to determine whether a patient has a specific signature? That's the story I'm going to tell you. One of the signatures, which happens to be the third one, signature 3, turns out to be a manifestation of homologous recombination deficiency, which is really important for clinical application. Okay, so we'll come to that. Okay, are there signatures for other types of mutations? By that I mean copy number and structure variations and so on. And we have another project where we're trying to identify this from the blood. Okay. Okay, so I'll tell you a little bit about this package. And I can't tell you all the details of it, but I'll tell you the problem that it's trying to solve. Okay, so just for some of you who are a little bit more mathematical, let me just tell you a statistical interpretation of what we're trying to do here. The data that we get is, suppose you have 3 different signatures going on in your sample, and then whatever observations that you make would be some combination of those signatures. So we represent that in some n-dimensional space like this, okay? And my matrix factorization is trying to find some cone that encloses this, right? So this is, if you were given what these signatures are, this is the data that you generate, but you don't actually know what those signatures ABC are. That's what you're trying to infer. And the matrix operation, you will try to solve this. And it turns out that there's a non-uniqueness problem, right? There are many solutions that will explain this, okay? And that's a problem because later on, I'll show you an example where given a signature, we're trying to find whether that patient has a particular process. And if the solution turns out to be non-unique, that's a problem, right? And so there are lots of possible solutions, and NMF as an algorithm is an iterative algorithm with many initial starts. So you do it many times, thousands of times, and then take some sort of average. Okay. Well, it turns out that this could lead to misidentification, okay, or false signatures associated. Okay. And so one proposal is to use another variant of NMF called minimum volume NMF. Okay, where we impose additional criteria to make it more unique. Okay, so let me just illustrate the problem. Okay, so we do an experiment where suppose there are 4 signatures, 3, 8, 12, 23, and we just simulate some random combination of those. Okay, and so if I simulate those, I find these little dots in some PCA space. Okay. And now what we try to do is solve using this NMF. And in this particular case, I got 4 different components. So it actually did a pretty good job. It recovered, if I have enough of these samples, it recovered 4 components. But it turns out that this is pretty similar. The cosine similarity of this to the original signature is pretty good. And this is pretty good. That's pretty good. This one, not as good, okay? Yeah, it's not exactly like this. And so when I try to do a signature assignment, okay, which is what you would have to do if you're given a sample and you try to figure out what the components are, match the catalog, this is actually a sum of these 3 different signatures, okay? And so what happened here? This other thing, SPS 39, is needed to come up with some combination that will explain this. And so using a standard algorithm, you will say, okay, it's made up of 8, 12, 23, and 3, 39, and 23. So this 39 just got into this catalog or for that case by the fact that this matrix process is not perfect. Okay, And so we can try to solve this using this other approach, and you actually don't have that problem. All I'm trying to, the bigger point that I'm trying to convey is that this process of matrix inversion is not perfect, and you can introduce errors, right? And this becomes really important because of the signature 3 example and other cases where for a specific case, we want to know whether that case, the mutations observed in the case was generated by this other mechanism, okay? Just to give you another example of this, there was apparently a lawsuit where the claim was that this individual was living near a factory that produces certain chemicals. And the individual says, oh, I got my cancer because of this chemical. How do you prove this case, right? If you can associate the effect of that chemical in a particular mutational signature, and you find that signature in that person's DNA, that is a sure way to do it, okay? In fact, I was once called to jury duty. It was a smoking case. And I was trying to get out of that because my wife would shoot me if I had to be in that jury for 2 months. They said it would take 2 months. It was involving Philip Morris. Lots of, high stakes. And I said, did you sequence the tumor and try to calculate mutational signatures? And that was a way to get out of this. Okay, because I said, well, unless you can put, because you can actually have lung cancer without smoking, right? The fact that because there's about a quarter of cases with non-smoking related lung cancers, right? How do you show that it was caused by smoking? Okay. All right. Another thing is that the catalog keeps changing because this process of identifying the signatures accurately requires a lot of data. And so here, there's a well-studied enzyme, APOBAC, that's really important. We're trying to figure out what that signature looks like. In 2013, with 500 whole genomes, it figured out that there were 2 processes that look kind of similar that's attributed to APOBAC. Well, it turns out that as we got more and more data, the signatures kept changing, and now it looks like this, okay? This is, in fact, the correct one, okay? APOBAC will generate, actually has 2 kind of branches, 2 mechanisms through which it generates mutations, and they correspond to exactly this, okay? So they did get it right, okay? For UV light, initially there was 1 signature, and now there are 4 signatures, okay? Exactly what the different components are, that's what people are trying to figure out, okay? But I mean, could we have, if we have 100,000 genomes, are we gonna have 17 components? Who knows, right? And so that's a mathematical question. And so here's an example of what we try to do here. So this is a signature derived from experiments. So in this paper, they actually took some organoids and put this strain of E. coli that will generate this, has this genotoxic effect and generate certain mutations. And from experiments, this is what you get, okay? So somehow this mutagen has that preference for the sequence, okay? And so you can actually recover this pretty well from 500 colorectal cancers, okay? But you have to have 500 colorectal cancers. So before, when there was a big consortium that I was part of that had a bunch of papers derived from our analysis of 2,700 genomes. Unfortunately, it only had 50 colorectal cancers. So then we could not come up with a signature. The best signature that we could come up with was that for colorectal cancers. Well, it turns out that using another method, okay, that we have developed, even with 50 colorectal cancers, we think we can find a signature that looks very much like that, okay? So this is where a good computational method will allow you to identify the signature even if you don't have hundreds or thousands of cases, okay? And so this will be incredibly economical if your goal is to identify signature associates with a particular mutagen. Okay, so I'm not going to tell you the details of how that works, except to say that the assumption that standard methods make is that whatever processes occur are occurring independently in different samples, and that's clearly not true. And so there's some fancy embedding method that we can use to obviate that issue. Okay. All right, so that's a lot of methodology. So if you're more interested in applications, that was probably a little bit too detailed, but I just want to illustrate the fact that this is really a growing field. Some of the signatures are incorrect, and we really want to have a more complete catalog. And so, there's a lot of effort where you're trying to basically try all the mutagens that you can think of, and then sequence the whole genome and generate a catalog, and then trying to match that. And because the catalog is changing, different papers are referring to different versions, it's a bit of a mess out there, okay? Now, I do want to tell you a more practical story, as I promised, okay? The story of signature 3, okay? And then the perils of translational science. I was meeting with a bunch of people today, and they were asking me, well, you were studying math, and why did you come to do biology? I told them I did all I can to stay away from biology until I got to my postdoc. And then there are some interesting things that I found then. But my big motivation was to do something useful, right? I didn't want to just do fun problems. I wanted to apply it to solve some real problems. And I'll tell you, my somewhat successful, but not quite effort on this. Okay, so signature 3 is a homologous recombination deficiency signature, okay? Now, it's kind of hard to work with signature 3 because it's kind of flat. So anytime you have a vector that's kind of flat, it's just hard to find it, right? Okay, how do we know that this signature corresponds to homologous recombination deficiency? Okay, there's a lot of proof for this. You can actually do experiments, right? You can knock out BRCA1, RAD51C, PALB2. You can knock these out and then sequence the whole genome. And guess what? They all seem to generate a very similar signature. Okay, so we feel pretty good that this signature 3 is an indication for homologous recombination deficiency. Okay. Now, why is that important? Because there are a class of drugs called PARP inhibitors. Okay, relatively new, very effective. Some people will say this is one of the major class of drugs that have been successful recently. There are lots of trials going on. I'm sure this number is much higher now. Without telling you all the details of how this drug works, it's been really successful in breast cancer and ovarian cancer, and potentially in other cancers, but there are lots of trials to find out. Now, in 2017, there was this paper from Sanger where they tried to use whole genome data to identify which individual has signature 3, okay? And they could actually come up with a pretty good classifier for this, okay? So they said, okay, this is their algorithm. You could just use rearrangement or substitutions or all those things. But in fact, if you combine them, you get the best predictor, okay? Well, it turns out signature 3 or HRD will generate, certain tandem duplications and so on. So it's not just the point mutations that you observe. Now, why are we doing this? Because why don't we just look for, you could say, why don't we just look for mutations in known genes, right? Why don't we look for BRCA1, BRCA2 mutations, and so on? That's in the homologous recombination pathway. Well, that's because a lot of these cases that are hard to detect because the genes are not known to us, or the genes are dysregulated for other reasons, right? You could have a methylation of BRCA1. So unless you have a separate assay for that, it may not have a point mutation, right? So it turns out that there are a lot of cancer genomes that do not have the mutations in the well-known homologous recombination pathway that still have HRD. And those are the cases that we want to identify. So we don't actually care exactly what component was deactivated. The consequence is that the mutations are of the signature 3 type, right? So these are all different kinds of mechanisms, epigenetic silence, PTEN loss, et cetera, okay? We don't care. All of these things will generate signature 3. That's why it's more efficient than trying to do multiple assays or try to find a comprehensive list of genes. And so our goal is to comprehensively identify HRD cases that will increase the number of people that will benefit from PARP inhibitors, okay? Because we have this amazing drug. We just need to who to give it to, okay? Now, given this 2017 paper, the first thing you can think of was, well, we should just do whole genome sequencing in all these cases, right? Okay, and I thought, hey, we can just have whole genome data, and then we'll identify these. And so this is how some of us computational biologists think, right? Well, why don't they just sequence whole genomes in the clinic, okay? Of course, the reaction that you get is not, wow, that's amazing, okay? Your problem is solved. We'll implement it in the clinic, we're done. The reaction is, okay, why, right? It's not that easy for a clinician to do whole genome sequencing and apply these algorithms, right? And so whole genome sequencing is nice. I mean, we love it because we can learn lots of new things from it. And it's actually not the cost, right? It used to be very expensive. Now we do it for about $350, okay? I'm sure you can send your samples out and get it for the same price. But it generates a lot of data, right? And people don't appreciate how complicated the downstream part is. A lot of data, okay? Expensive to store and analyze data. We have thousands of genomes in my laboratory. Just the cost of that is quite a bit. Just storing, if you just actually calculate the money that I would have to pay if I were to move all my data to Amazon, it's hundreds of thousands of dollars just to store. Forget compute, okay? So it's not that easy. It's hard to find analyst experts, right? I mean, some of you know this well, but a clinician sometimes thinks, okay, I'll just hire some summer job for an undergrad and he can figure this out. It's not that simple, right? It actually takes some specialty to figure out how to identify these variants and so on. And interpretation is not straightforward, okay? And so I thought, okay, this is too idealistic, okay? They're not gonna go and do whole genome sequencing. Cost, time, expertise that I just mentioned. How can we do something a little bit more practical? And I thought, okay, what about we do this from panel data? Because there are a lot of academic centers that are doing panel sequencing, okay? So could we apply the same signature detection algorithm from panel data? It turns out that the computational part of this is quite challenging because you don't have that many mutations, right? So here is signature 3 from whole genome data. If I have a panel, instead of this spectrum, this is what I get, okay? You get like 20 mutations in the extreme case. How many mutations do you need for you to be able to call in a clinical case? Okay, some people will say, I mean, you need like thousands of mutations. 100 mutations? No. Okay, in fact, I thought it was actually quite difficult, maybe not doable, okay? And then Doa came to my lab. She was a MIT high energy physics PhD who wanted to do something in the biological sciences. And so she left CERN to come to my lab. And within 3 months, she figured out how to do this. I was surprised at how well it could work, okay? Basically, from a very small number of mutations, we can do not as good as whole genome, but reasonably well, okay? Now, how do you do this? The trick is you are trying to take advantage of all the whole genome data that have been accumulated. Okay, it turns out that for certain tumor types, we know which signatures are more popular, which signatures occur together, okay? And we use that information to help us make accurate guesses for these panel sequencing data. Without that, we just wouldn't be able to do it, okay? And so the more data we have, so we're now trying to look at the Genomics of England whole genome data sets, 100,000 plus, to see whether we can further improve that. But that prior knowledge is critical. Okay, so what do we find? Well, so we find that in the triple negative breast cancers and ovarian cancers, only a small fraction are actually BRCA1 or 2 cases, okay? The majority are actually BRCA wild type, but they still show HRD signature, right? So if you just give PARP inhibitors to BRCA1 mutant cases, you're only doing it to 30% of the cases. You're missing the 70%. And in fact, we do this for lots of different tumor types, and you see that the percentages are small, but not so small, okay? And so I would talk to a pancreatic researcher who says, Peter, this is great because I wanted to give PARP inhibitors my cases, but I don't know which 7% have signature 3, okay? And maybe with your tool, we can figure out what those, because they were getting these panel data anyway, right? So it's no cost to them, it's just additional analysis. There is, in fact, a commercial test for this. Biogenetics sells something called My Choice CDX, $4,500. So a lot of clinicians actually send out this for $4,500, okay? And our proposal was it's free, just a little compute, okay? Now, I thought that we have to do a lot of work to ensure that it's almost as good as this commercial test. And in fact, we spent a lot of time trying to work with clinical trial data to make that case. And so I'm going to go through this really quickly just to show that, in fact, we have tried. So here, in our original study, we showed that BRCA wild type, but signature 3 positive cases have a similar survival profile as BRCA mutant cases, okay? So they're BRCA wild type, but if they have signature 3, they show similar behavior. And we do this for many cancer types. In some clinical trial data with colleagues here at the Brigham and Dana-Farber, we show that signature 3 is actually a good classifier. We compared to lots of these other standard tests out there. And so it was some work to find samples where we had these other data types. In this particular case, Mir and My Choice did not separate the cases well, but we actually did a little bit better. There are other cases, trials, we also show that this works, okay? And in fact, in this case, this is a signature 3 positive cases with panel data. It turns out that BRCA is involved, but many of them, at least half of those, are actually methylation cases, okay? So BRCA does not have a point mutation, but it's methylated, and you would not have known, right, if you look for point mutations. So all this to say that to convince clinicians, you can't just say, oh, here's my algorithm, okay? Go and read it and then do your thing, okay? You actually have to demonstrate for their data, right? So it's a lot of work. Now, but still, it was not enough, okay? So after we published this paper, we actually did some reach out to companies, and certain companies, you can imagine that if you're a pharma company making PARP inhibitors, you do want to identify these other cases, right? And so we had some license deals, but most of it was just very difficult, okay? Why was it difficult? They'll say, oh, Peter, that's great. I read this paper. It looks like it's reapplicable, but I don't have a software engineer. Can you help? Okay? Say, hey, did you test it on my cancer type? I have a different panel. Can you optimize for my panel? Hey, I get an error message. Can you help? Okay? Usually not related to my algorithm. It's like something about their system, right? Well, they don't know what to do because they don't have a team that can work on this. Can you rewrite a different language? Whatever, right? All sorts of things. We just could not. I mean, we're getting queries from academic centers all around the world. We just didn't have the bandwidth to do it. And of course, it's hard to get a postdoc to do this because it's not going to necessarily lead to a paper, certainly not a high-profile paper, right? A lot of tuning and customization. And so in the end, I was like, okay, I don't know what to do. Okay, I wanted to do something good, right? And it was really difficult. So I'm still working on it. We do have a small internal grant now to translate this, and we're hiring people to basically test this for different scenarios, right? Every medical center has to have their own panel, an ego thing, right? Okay, and so you got to do it. Some companies like Myriad Genetics or Foundation Medicine, they have something like this, okay? But it's actually, for them, it's also very difficult to modify their pipeline because it means that they have to go through the whole approval process again. So it's not that straightforward, okay? Now, just to give you another point of frustration. It turns out that the hardest part for me in this process was actually not the development of algorithms, okay? We try to work with Harvard Affiliated Hospital, literally a block away from my office, okay? And they were doing panel sequence project. They had 100,000 panels, okay? And so a lot of physicians and scientists say, hey, this is perfect. Can you help? I'll say, okay, I'll find some people to work with you. And the physician scientist says, I would love to share the data with you. Okay. Then the lawyers come in, okay? The raw data cannot leave the hospital, okay? Now, it sounds like at Michigan, you don't have that problem because you're all within the same, domain. But for most hospitals, this is, I think, a relatively new thing, but they're extremely protective. Literally, data cannot be moved. And so the solution that they came up with is, oh, we have a team, okay? We will run it, give you the result. You tell me how to modify it. I will modify it, run it, and give you back the result. And most the computational students know that this is not a way to do things, right? And in fact, later on, they said, we're too busy, can't help you, okay? The extent to which I have gone up at the hospital chain, administrative chain to resolve this problem was more than anybody should do. I still could not resolve it, okay? And this is a real problem. And this is just a bottleneck that I had not anticipated, right? Everybody wants to share the data and do things, but the legal part is not straightforward. And finally, I had the idea, oh, maybe they can hire me as a postdoc or something, okay? Well, that didn't quite happen, but I got my postdoc in my lab to have some joint appointment at this cancer hospital. That was the only way where we could get access to data, okay? Of course, in retrospect, this is what I should have done earlier, but I can't get an appointment at every hospital that we want to help, right? So I don't know how to solve this problem. Now, some of you don't have to deal with this, but I deal with this all the time. We have a big group, lots of projects, a lot of that involving public data, right? Published data. Turns out that data sharing agreements are extremely hard. Some of the PIs out here are probably, yes, I know, because I had to do this. I probably have 30, 40 agreements with various places. Each one is just a complete headache, okay? And it's because of just legal offices being very risk averse, okay? And genomic data privacy is important, but I've been filling out forms for the last 20 years. Nothing has happened, but they want to make things harder, right? Because they want to make it safer, okay? And so this is kind of a strange time. Well, we're living a strange time for many reasons, but this is one aspect, okay? And it's really unfortunate because I want to do good things. I'm willing to invest in my people. We just can't do it because of the legal problems. And so hopefully other people, can solve this problem, okay? Now, so some lessons I've learned, well, writing paper is the easy part, actually. Method should be, but for the students out here, right? You have to address practical questions, I think, okay? You can have your kind of fun, kind of dreamy project, purely scientific, that's important too. But for some of you that are really interested in translational aspect, right? Rather than saying, publishing a paper and saying, oh, it could be translated by somebody, right? You actually want to be there trying to test this with collaborators, but the implementation takes a long time. Okay. All right. So that's my story. This is to say, I didn't actually talk about all the amazing collaborations that led to lots of interesting science and interesting clinical applications, but I did want to just share that things are not as simple and straightforward as one might expect. But it is really important for us to develop methods that are practical and then spend the time and the energy. Just for the last few minutes, I wanted to share this. Do you know this paper, Mark? Oh, okay. I actually give an hour talk on this topic, but I'll just give you a 5-minute version, okay? I was so impressed by this paper, and you'll see why, okay? 1957, William Shockley, okay, on the statistics of individual variations of productivity in research laboratories, okay? You can just Google this and find the PDF. Okay, he's a famous person. He invented semiconductors and led a very successful group at Bell Labs. Okay, as many of you know, Bell Labs was this kind of amazing laboratory for decades. Invented many of the things that we use in our daily lives. Okay, if you read the paper, it actually starts with something that I thought was kind of funny. The following page is winner of the Nobel Prize in Physics presenting novel study of today's most precious commodities, scientific productivity. Okay, so this is a long time ago, but still, some things are timeless. Okay, now if you read about him, it's not flattering description. He may be the worst manager in the history of electronics. Autocratic, domineering, erratic, hard to please, and increasingly paranoid. Okay, so unlike any PIs that I've met today, but there are such researchers out there. But he had incredible insights. Okay, and that's what I want to share with you. Okay, now the question is just how is it that some people are so much more productive than others in research, right? If you look at physical characteristics, okay, there's actually very little difference, right? I mean, you get the height variation, and it's not like there are 2 feet tall people and 10 feet tall people. There's a range, right? Racing, I looked up some times, 100-meter sprint. I mean, we're talking about the best runners, right? We're 0.1, 0.01 second differences, right? Our physical, characteristics do not vary as much. But somehow, research productivity varies, right? All right, so what Shockley did was look at 160 people from Los Alamos Laboratory. This is a famous research laboratory in New Mexico. And he looked at the publications per person. Turns out that most people didn't publish anything. There's one person that published 12 papers and so on. Okay, this is not a normal distribution. Okay, why isn't it normal? Okay, research productivity appears to follow a log normal distribution. Okay, that means if you take log, it looks more like a normal, right? But it's not normal. Why? Okay. Okay, and he actually looks at some other data sets and finds similar distribution. Okay, and so he has one explanation that I thought was kind of entertaining. Why log normal distribution? Suppose that some attribute of the human brain allows individual to be aware of M ideas, okay? And their relationships. Their relationships is the important part here, okay? So for instance, if you are designing a self-starter, you actually had to put together multiple ideas, okay? I don't know if you know what a self-starter is, but in the old days, you have to crank the motor manually to get your engine started, right? But now we don't have to do that, right? That's because someone thought, okay, maybe there is a different way to do it, and that maybe you can store the energy in a battery, okay? Maybe the way to do this is have a small motor spinning at a very high speed to get it going, and then disengage so that the engine, the pistons will start, okay? So that actually is a combination of 4 ideas that I just listed, okay? And so you need to be able to have a certain number of ideas and then mix the combinations, right? And so he says, okay, if you happen to be an M3 person, like your brain only holds 3 ideas, you could not have come up with a self-starter. But if you're an M5 person, there are actually 5 permutations of 4 ideas that you can hold in your head. Okay, maybe you can see where I'm going, right? If you have to have the M equals 6 brain, there are 15 ways to rearrange a combination of 4. Okay, so this might be linear, but the impact is nonlinear, right? Your combination increases much faster, right? And so that's his explanation, right? Our brain capacity might be linear in some way, but the combination might be nonlinear. Okay, do you find that interesting? Okay, maybe, okay? All right, and so you can actually have 50% increase in brain capacity, but you can actually have 15-fold increase for the likelihood of coming up with that idea. Okay, well, how does this work? He says, in terms of your productivity, there are many factors involving publishing a paper. It's actually interaction of many factors. Okay, you need ability to think of a good problem, ability to work on it, okay? Ability to recognize a worthwhile result, right? Sometimes we get some result, but we can't tell whether it's worthwhile or not. You keep going, right? So you need to know when to stop. Ability to make a decision as to when to stop and write up the result, okay? And you need to listen to your PI for this. Ability to write adequately, right? Critical skill, right? Ability to profit constructively from criticism, right? This is an incredibly valuable skill. Determination to submit the paper to a journal, right? Some of you are probably thinking, oh, that's me, okay? Persistence in making changes as a result of journal action, right? Sometimes you get the review back, oh, I don't want to think about it, okay? Months go by, okay? This is another important thing that he recognizes, and we recognize, we just don't think in these terms. Now, he thinks that this is actually a product of these factors, okay? It's bad news because the factors are multiplicative, okay? If you don't know, if you get 0 on any of these factors, you're done, okay? True, right? If you don't recognize, if you don't know when you have a good result, your productivity is 0. If you cannot bring yourself to write it up and submit it, your productivity is 0 and so on. It's a terrible news, okay? But the whole point of this, it's also good news, okay? Why is it good news? Because the factors are multiplicative. It means you can just be a little bit better in each category to have a high throughput, high output, okay? So I did some calculations. If you're 10% better on each of these categories, your output will be twice as much. If you are 50% better in each categories, you will be 25 times more productive, okay? Why am I saying this? Because some people, some trainees that I talk to, okay, I've got to work harder. Maybe if I work twice as hard, which is very difficult, right? Maybe I'll be twice as productive. Sometimes that's not it, right? You've got to work on these other factors that will increase your P, okay? And not focus on one, okay? And you don't always have to have the best ideas, right? Because there are plenty of ideas out there. You need to work on these other things. So, and so the way to improve your productivity is actually trying to focus on the things that you need to increase this final P. And I can tell you from my experience that different trainees work on different areas of this, or different PIs need to work on different areas. And just the final bit that you might find interesting, find interesting, he looks at salary versus productivity, and he says there are many factors that go into compensation, but on average, an increase of 30 to 50% of productivity is necessary for an individual to obtain a 10% increase in salary, okay? And so all this to say, this is a fascinating paper, and he didn't have a big data. He just had a tiny data set, 70 years ago, and he probably came to the same conclusions that some of us would with a much larger data set. Okay, so that was just a fun thing to for the trainees, and so I have a lot of people to thank. Post-COVID, I took my whole lab to Florida. The only problem was that they expect that every year now, so we'll do that again sometime. But a lot of wonderful people, a lot of collaborators, both in Boston and elsewhere. So thank you for listening. Lots of questions.
Great, thank you for the wonderful talk. Any questions from the trainees first? Yes, Changbin.
Thank you for a nice talk. I'm just curious for your HRD story. So do you believe your HRD estimator a type of medical device or a part of new drug regarding FDA approval, if any?
Can you just say one more time? Sorry.
I mean, when regarding the FDA approval.
FDA approval.
Yeah, there are two types: medical device or new drug. Do you believe? Yeah.
I have no idea. That process needs to be taken up by a company, right? And my understanding is that it's actually a process, right? Like you don't get an approval for an algorithm, you get approval for your entire pipeline, right? And once it's approved, it's fixed, right? And so we basically need I need a company who will incorporate this into pipeline. But that's much further down. I mean, I want this to be used in an academic setting, and lots of good papers come out because they discover these individuals that have HRD. And then I think we can worry about the next steps. And so, I'm surprised that a lot of companies are, not as many companies are interested. And part of this, again, is just they have their own niche. And sometimes I'm finding that the industry is really driven not by the need or the desire to the greatest good for the greatest number of people. They're looking at markets, and sometimes maybe they don't want to identify the 5%. They want to get an approval for 50%, right? Then their market is 10 times higher, right? So it's a complicated business, as I'm learning. Yeah.
Thank you.
So I'm interested in the method you are talking about, the signature analysis. So I think maybe for some gene, for some tumor type, you have more complicated composition of the signature. For some gene, it might be more simple. And I think the ability of the sequencing data to interpret those signatures is kind of depends on the complexity and also the resolution of your sequencing data. So I think maybe we can design an algorithm or method to determine what kind of sequencing data, maybe coverage or sequencing accuracy you need for specific genes, for specific regions that composite with specific signature. Right. Yeah.
Sure. So different tumor types obviously are variable in terms of the difficulty and the complexity, right? Obviously, if there are lots of mutations, it becomes much easier, right? Melanoma is easy, right? Rhabdoid tumors, they're very quiet genomes, very hard, okay? And so the power to identify those are variable. And power to identify specific signatures is also variable, right? Some signatures are easier. And so there's actually a lot of kind of manual work in trying to figure out how to approach this problem. So you can imagine like MSI tumors or other hypermutated tumors, they can actually sort of contribute a lot more to your analysis, and sometimes we have to manually take those out. There are lots of cases where you actually need to understand a lot about the tumor to decide whether the result is correct or not. And so there's just a lot of back and forth. Does this make sense? If not, maybe we need to modify this part of the algorithm. So it's not as straightforward as, press a button and this is the final result. And so I think that's the case now, and it will always be the case. So it does require kind of interaction and knowledge of this in driving this, because there are parameters that you have to tune. Okay.
One last question. Now faculty can ask questions.
Okay, yeah, go ahead.
All right. So you mentioned the FDA, that you would want your algorithm to be used in an academic laboratory, a hospital laboratory. But laboratory developed tests are subject probably in the near future also to FDA oversight. Especially for computational algorithms, the FDA has released guidance on how such computational algorithms should be validated. And it's actually quite elaborate and almost, one could say, extreme. For example, algorithms need to be tested that they perform well or equally well in different ancestries, which obviously in genetics is a challenge. So my question is, do you think that your work that is nicely validated in publications should further be scrutinized by the FDA before it is implemented clinically?
Well, we'll see how things shift. I mean, we take flu vaccines that are untested, right, every year, right? I mean, we don't do clinical trials every year for different strains. And so somehow we have slightly different standards. I mean, I think the I think the best way to do, I mean, just because we all have limited amount of time, and like in your case, right? I was talking to Martin earlier today. I mean, he's got this data set from the hospital system here, like very similar to the panel sequencing, actually much better than the panel sequencing that we were working with. And so there's a lot of opportunity there. And it's just really energy that you have to put in to take it to the next level. And so, as an academic, do you have the energy and time to do it? Who's going to fund this? And so I think at some point it needs to be a company, and hopefully they know how to do this better. And so, yeah, I mean, if I had a lot more time and energy, I'd probably start a company and try to do this. Actually, we have tried to work with companies. Some company that has hundreds of thousands of panel data, and data access agreement is where we got stuck. Okay, it took 2 years. Lawyers. Okay, our lawyers want X, such and such benefit if it does, X many dollars from their assays. Their lawyers. That's where it got stuck. After 2 years, I've given up, and so I just a sad state of affairs at the moment.
I have one more question.
That's not a question, it's just a comment. First of all, thank you very much for being here again. I think Martin's question is really about the business world. The reason the FDA is empowered by Congress to put requirements like this on testing, and they're reluctant to do post-marketing surveillance because they don't actually have an authorization from Congress to do that or mandate it of the companies. It's all in the lobbying and the positioning of companies to see if they got something that is selling, they can keep others out or get the benefit, as you were just describing, maximal participating selectively. That's a complicated statement about our economy and our society.
Act which is stuck in the last 3 or 4 years, which should have.
Thank you so much for Gil's wonderful comments. That's a great conclusion of our talk today. And please join me thank Peter's wonderful talk today.
Thank you.